Okay.
Okay.
So we start the presentations at 10:05 or 11:05 rather, which is when MIT classes start. Okay, for whatever that's worth. So is that the only AWG you have?
No, no, we have a couple. We had a very cheap one when we started that cost us less than $10,000, and now
Chase Scientific.
No, Wave Pond.
Well, that's Chase Scientific, yeah.
And we also have the Bruker spin jet.
Mm-hmm.
That integrates nicely with the spectrometer. It's good for folks who are not that instrumentation centered in their work.
Yeah, yeah, yeah. 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 Well, I mean, the we have we have three of those wave ponds, and actually one of them just died, the one we use for a pulse programmer on our 140 gigahertz EPR. And it turns out the Chase Scientific is not making them anymore, and they're actually so we just ordered one off eBay. So, well, I mean, you know, Dave Rubin we'd have to build one from scratch, which would be a, you know, that would be a lot of work. Yeah. So, so we'll see what happens with it. All right. Hopefully it'll work. And if it doesn't work, well, then we have to. You don't happen to have a spare, do you? An extra?
No, I wish I had.
Uh-huh. Yeah. There's probably some simple component on the thing, you know, but if you replaced one chip, it would probably be fine. You know, but we can't get Mr. Chase to talk to us about that, right?
Bob, do you know like which component broke? Because like he has been there for ages, more than 10 years, and it was working fine. And I'm just curious, like how did it break? Or...
It's the PCI slot. Is that right?
Yeah, it's a PCI slot. Yeah.
Yeah.
In the computer.
Yeah. Mm-hmm. So that's what we talked to Mr. Chase about this.
Yeah.
He said it's likely that. He said that's what always fails. He said, but we just don't have any more of them.
Mm-hmm. If I'm not mistaken, I think like that model was chosen because like it's compatible with Spellman. And maybe you can ask Boris if he has like other suggestions to replace the card.
Well, as I said, we replaced we ordered a card.
Okay, good.
There are several available in China. So we ordered one yesterday. Yeah.
The same one, the same model.
It's the same model. Yeah.
Okay.
All right.
So
when do we start?
Well, 2 more minutes, 1.5 minutes. Okay.
Okay.
Get going. Okay. And then Kong, you want to do your introduction now? What's your...
Yeah, sure.
Yeah.
Let me just. Okay, hello everyone. So I would do a very short introduction because I've been doing this on and off for almost 2 years now, because we started exactly almost 2 years ago. Okay, so for your information, I put up the mailing list and the past recordings in the chat room. So if you are interested in watching some of the past presentation, you can go to the Griffin Group website. I regularly update the information there. So today we are quite delighted that we have two excellent speakers here. So the first speaker will be Stefan Stoll, and the second speaker is Rachel Martin. So I will just do the first introduction. So I'm sure most of you know Stefan because like he made a very famous software, EasySpin, which I benefited a lot. So by just reading through his biography, so he, of course, his lab maintains and develops EasySpin, and he got a silver medal for methods from the International EPR Society. He's originally from Italy, and he did his undergrad in University of Technology in Austria. After that, he went on to start his PhD with Arthur Schweiger, you know, the one who wrote the famous EPR Pulse EPR book. Then after that, he started his postdoc with Dave Britt in UC Davis, and eventually then he got a professorship in the University of Washington. So we are looking forward to listen to his talk, which the title is The Mechanism of Electron Spin Decoherence. So the floor is yours, Stefan.
Thank you very much, and thank you for this kind invitation. It's great to be able to present in front of this crowd. Today, I would like to present a topic that my lab has gotten interested in the last 2 or 2.5 years out of a practical necessity. So it is understanding the coherence dynamics of electron spins. So first of all, why does decoherence matter? And the angle from which we were coming from is that our lab is heavily involved in using a form of EPR spectroscopy called DEAR to measure distances between spin labels on proteins. And then from these, through their dipole interaction and through the resolved distance distributions between those spins, resolving energetic and geometric details about the conformational landscapes of these proteins. The spin labels we use for this purpose are mostly nitroxide radicals. Here you can see the most famous one called MTSL. And the key is if you want to access long distances, you need to be able to keep the coherences on these electron spins alive for a long time in excess of 10 microseconds. Another area where electron spin decoherence has gained has increasing interest is in quantum science. There's a lot of folks in the chemistry community who are building spin qubits for potential applications in quantum sensing. Of course, the solid state physics community has been working with the Diamond NV- Center for a long time. It's kind of a carbon-based radical, but there's also transition metals and rare earth spin centers that have long decoherence times. So understanding decoherence is something that benefits multiple fields. And our angle is mostly coming from seeing if we can get some understand decoherence to extend distance ranges in DEAR spectroscopy. So 2 of my students started asking the question, what is the origin of this decoherence? And actually, is there a way to quantitatively predict the decoherence from just knowing the molecular and solvation structure of an organic radical? Sam did he is a fantastic theoretical physicist. He did all the theory work, and Lizzie, as a biochemist, she did the experimental work. So what we focused on and trying to understand decoherence was first a very, very simple nitroxide radical. We'll put it in a dilute, take it per deuterated, put it in a dilute solution, and freeze it and look at it at below 50 Kelvin. This has the advantage of isolating one of the many contributions mechanisms to decoherence. So first of all, T1 is very long. So T1 has no effect on decoherence. There's minimal thermal motion second. So we don't get any thermally relaxation. Third, since we're dilute, instantaneous diffusion is minimized. And fourth, through perdeuterating methyl groups, we quench methyl tunneling contributions. Of course, we're not the only ones to ask these questions and look into like what's going on. There's been work from Russian groups in the past, Gareth and Sandra Eaton, Marina Kvider, and also from Gunnar Jeschke's lab. So the simplest manifestation of electron spin decoherence is, of course, the Hahn echo, where you can, as you increase, here's the sequence, the total pulse length is 2 tau, and as you increase the total pulse length, the spin echo amplitude decreases. And many people over the past decades have measured these decays and noticed that it usually takes the form of a stretched exponential decay, in addition to some wiggles that have separate reasons. The stretched exponential decay is the time constant of this decay is called the phase memory time or decoherence time or coherence time, depending on which literature you read. And then there is a stretching exponent psi. The stretching exponent has values between 1 and 3 typically, 1 being just an exponential, 2 being a Gaussian, and 3 some sort of like hyper Gaussian or super Gaussian. In the systems we're looking at, the stretching exponents would typically be around between 2 and 3. So this is a phenomenological equation, and we wanted to understand the actual physics behind, and then see if we can use the understanding of this physics to improve the coherence times in our deer spectroscopy systems. So back to this simple nitroxide radical. It's embedded in a water glycerol solution. Here I'm just showing the water, and what we're set out to do theoretically is just basically to simulate the spin dynamics of this entire system, which is one electron spin here at the center, surrounded by a bath of hundreds of proton spins, and we need to go up to beyond 1,000 protons to converge calculations. Basically, from a magnetic point of view, we have one red dot with electron spin, one blue dot for the nitrogen 14 spin, and thousands of protons. Now, of course, in terms of spin dynamics, this is an intractable problem. So after we've generated the structure of the molecule using density functional theory, we've solvated it using molecular dynamics, we end up with a three-dimensional structure that has a spin Hilbert space of 10 to the 300 states. Totally impossible. Now, people, especially in the solid state community, have developed a range of methods to deal with this problem, to cut this space down. We are using a method called cluster correlation expansion, which was developed by Renbao Liu about 14 years ago. And it consists of the idea that the whole spin dynamics problem in terms of like density matrix propagation, the signal can be written as a product over clusters of overlapping clusters from spin pairs. And you can see that here on the right-hand side, you can see the electron spin in the center. You can see a cluster of 1 nucleus. You can see a cluster of 2 nuclei, or 3 nuclei and 4 nuclei. And refer to these as like 2 clusters, 3 clusters, and 4 clusters. This cluster correlation expansion is a product. And if you increase the size of the clusters, in the situation we're looking at, the contributions decrease with cluster size. In a way, this is a perturbative expansion in the dipolar coupling between the nuclear spins. So if we do this and we truncate the cluster size 2, 3, or 4 and put some upper limit on the nuclear-nuclear coupling, we can simulate these dynamics are approximated pretty well by including about 500,000 2-cluster signals and 200 million 3-cluster signals. It is hard, but it's feasible to do. This method is kind of the Hilbert space analog to the truncated state space, Liouville space methods, and that in NMR that pioneered by Emsley and especially by Ilya Kuprov. Kuprov's method, the more general, right? This is a very special case. We have only one strong magnet and a lot of weak magnets, and using this special structure of the spin system allows us to be a little more efficient than the methods implemented in Kuprov spinach. Now, here are the experimental results together with the simulations. On the left-hand side in black, you can see the experiments. And in red, you see the from first principles simulations without any fitting parameters. You can see a good coincidence, a good overlap between the experiment and the simulation. You can also see that we have a stretched exponential form of the decay. You can see that also in the case of the D18 tempo radical, we do get proton ECM effects. This is due to the pseudosecular hyperfine coupling between the electron and the deuterons, which disappears if you go to a proteated tempo radical, which then on the other hand shows a little bit of an interesting non-stretched exponential shape, which is due to the methyl tunneling that's of course activated here if you put back protons into the system. The third radical shown here is a triddle radical. So it's a carbon-centered radical with just an electron spin localized essentially on the carbon with basically no protons in a in the immediate neighborhood. and also the deuterons are very far out, which quenches ECM, quenches the methyl tunneling contribution. So you get a experimentally very, very clean exponential decay. Now, in order to get these red simulations, we only had to take this cluster correlation expansion up to second order, or include only pairs of 2 nuclei. Going further to 3 nuclei didn't yield any substantial improvement. And just a note here, this is all done at dilute concentrations at 20 Kelvin. This effect is can be observed at multiple frequencies. On the left-hand side, as I've shown you, this is measured at Q band, so 33 gigahertz, about 1.2 Tesla. At X band, 9.6 gigahertz, 0.33 Tesla, we can see this, the experimental manifestation is a little different. We get stronger ECM modulation, we get a more exponential looking decay, but overall, the simulations, again, very close to quantitatively reproduce the experimental observations. Now, okay, this is this is all good, but what can we learn about the physics? Now that this quantum dynamics model is validated, we can kind of break it apart and look kind of under the hood of what's going on. And one way we decided to go ahead is to define something called the differential coherence time reduction. So imagine the full system, where each proton participates in clusters that contribute to decoherence. What if we eliminate that single proton? Take, for example, here this water molecule. What if we eliminate this single proton? What would happen to the phase memory time? How much would it get longer? And we can basically do these calculations, basically dropping 1 proton at a time and rerunning the entire calculation to kind of get some blame factors for every single proton in the system. And here you can see a visualization of that. White indicating there's very little contribution to TM, and blue or black indicating has a very strong contribution. And you can see that some ring protons on the nitroxide radicals, for example, these 2 here have very weak contributions. But as you go a little further out, you can kind of see a ring where there's a higher concentration of dark protons, indicating that there is some optimal distance where protons in this matrix can contribute to decoherence. And we can break this apart and plot this in a more obvious way. So if on a horizontal axis here, you can see the distance from the electron spin center, and vertically, we are plotting these differential coherence time reduction as points here. And every point corresponds to 1 proton in this molecular dynamic structure. You can see the protons at the very close end, below 4 angstroms, they add about 10 nanoseconds per proton to the decoherence time, but this goes up to almost 30 nanoseconds if in a range between 5 and 7 angstroms. And after that, it decays pretty rapidly on a per proton contribution. But then, of course, you might say, well, this is per proton, but as you get further away, the number of protons grows with R squared. But then if you just do a cumulative TM calculation, shown here with the right vertical scale, you can see that the TM, as you include more and more protons away from the distance center, the TM starts to converge once you hit about 10 angstroms, or maybe say 12. So any proton beyond 12 angstroms in this kind of environment does not contribute to decoherence at all. So what is the actual spin physics here? And this is the slide for the theoreticians in the group. The spin Hamiltonian can be set in the high fields, in a high field situation with the 3 Zeeman terms for 1 electron and 2 protons. And we including the ZZ term of the hyperfine couplings for both electron proton pairs. And then importantly, we need to include the coupling between the protons, and specifically the flip-flop term. And in this 3-spin system, which is kind of the simplest system where this effect manifests, you can derive an analytical equation for the echo modulation, which has an interesting formula assigned to the 4th here, with a modulation depth and a modulation frequency. Now, this was already derived by the solid state community a while ago. Now, the modulation frequency, the modulation depth is depends on the different, here's a plot of the modulation depth against a particular quantity, which is the difference in hyperfine couplings, delta A, divided by the strength of the dipolar coupling between the 2 protons. And if that ratio is close to 1, the modulation depth is maximal. And the modulation is plotted here on top. And you can see this is kind of a modulation at frequency omega with a little bit of small second harmonic contribution on top. Basically, what you will notice, all these equations here is the complete absence of a magnetic field. So this is a field independent effect. It only depends on the difference of the hyperfine couplings and on the transverse field from these flip-flop terms. Okay, and for folks who don't look at spin Hamiltonians and product operators every day, here's a kind of a rough vector picture of how this how could think about this. Here's the electron spin. It sits in an external field, B0, and it also experiences a hyperfine field from the surrounding protons. Now let's switch over to the nucleus. The nucleus as well sits in external fields, providing Larmor frequency. It also feels the transverse field from a neighboring proton due to the flip-flop term, and it also experiences a hyperfine local field from the electron if it's in alpha state. So the effective field and the quantization axis of the nucleus is along this BF. Now, if you go into the other electron spin manifold, conceptually you think you just flip the electron spin, then what is going to happen to the electron for the nucleus is that of those three fields that determine the total local field, you only invert the hyperfine field. And as a consequence, the new effective field is at an angle to the old effective field. And as a consequence, we have some nuclear coherence. So there's precession. And of course, if the nucleus start precessing, the hyperfine field at the electron will become time dependent, and you'll get basically modulation of the electron. So coherence on one electron becomes coherence on all protons, and that modulates the frequency at the electron. That's kind of a rough vector picture of what's going on. So next we tried to look what happens if we change conditions in the matrix, and particularly when we fiddle with the proton concentration and the distribution of protons in the matrix. So first, proton concentration. Here on the left, you can see a series where we successively replace protons with deuterons. And it is well known that deuteration, of course, reduces the magnetic moment by a factor of 6.5, and therefore it prolongs the decoherence time. And you can see the experimental data here on top in a logarithmic scale for 2 tau vertically, and then here just on a normal standard scale. The Han-Eccles decay, 100% protons, you get a decay that's completely gone at 10 microseconds. With 100% deuterons, or actually, which is say 9 and 99%, that's what you can buy commercially. It almost stretches out to about 100 microseconds, so 10 times longer. On the bottom side, on the bottom part here, you can see the first principle many-body spin dynamics simulations, and they do a pretty decent job at capturing the time scales and also the shape of the experimentally observed decays. And again, the simulations are based on taking an hydroxide radical, geometry optimizing it with density functional theory, solvating it in a water and glycerol mixture using molecular dynamics, and then using basically density matrix propagation using cluster correlation expansion to just calculate the signal. We can fit these to stretched exponentials. And the two parameters, of course, are the phase memory time shown here on the top, and then the stretching exponent on the bottom as a function of proton concentration, right? So 100% proton, we would be about 110 molar. And at 99%, 100% deuterons or 99% deuterons, we're down to about 1 molar protons. And in red, you can see the experimental data. These are the TM fits to these data here. And the stretching exponents are shown here. They vary between 1.5 and 2.7. And then in the small black dots, you can see the simulations. And depending on, there's actually two versions here. One where we just outright neglect deuterons, and another version where we include the deuterons in the calculation, which is actually quite challenging. But in either way, deuterons don't really matter until you hit about like 2 molar protons, which is very, very dilute. What you can see is that the phase memory time has an interesting temperature concentration dependence. It's a proton concentration to the negative 2/3 basically. And the stretching exponent is also very concentration dependent. It goes all the way down to 1.5 at dilute proton concentrations. So where does this come from? If you would just think about a random distribution of protons in space, you can come up with a simple model that predicts that the phase memory time should go with the inverse of the proton concentration. Now, we decided to probe this a little more experimentally. And so in this water, we have a water and glycerol matrix. Glycerol is a glassing agent, but we can use this to our advantage because there's now two separate proton pools in this matrix. One is the exchangeable pool of the water protons and the OH protons on the glycerol, shown here in green or teal. And then there is the pool of non-exchangeable protons that are carbon attached. And there's always a group of 5 on each glycerol molecule. What we can do, we can change the proton concentration, these two pools separately, and see what the effect on the phase memory time is. On the right-hand side, you can see the result, the experimental data. On the bottom, the proton fraction is kind of mole fraction. On the top is the proton concentration, vertically plotted the phase memory time. And if you look in this area here, the red curve shows, or the red triangles show the data that I've shown before, where we have the same proton concentration in both pools. If we only put protons into the OH pool and leave the glycerol fully deuterated, the phase memory time is longer. Whereas if we put the deuterons, if you put protons in the OH pool, the phase memory time is longer. But if you have protons in a glycerol pool, then the phase memory time shortens by a factor of 2, even if we use the same bulk proton concentration. So obviously, it's not just the proton concentration that matters, but also the distribution of the protons. And one way just to look at the distribution is just to look at the radial distribution functions between protons. So the question is now, which of these proton pairs contribute to decoherence? Let's look at the two samples. First, the red one here. The red one is the sample where we have protons, most protons on the glycerol molecules in CH bonds. The pair distribution function has a peak at 1.7 angstroms corresponding to HCH. So geminal protons, but then there's also alpha and beta positions for proton pairs, peaks at between 2 and 3, and then at larger distances. If you go over to the OH sample where most protons are in the OH pool, we get a sharp peak at 1.5 angstroms corresponding to the intramolecular proton-proton distance. We also still see glycerol peaks, and that's because we modeled this with the purest deuterated glycerol commercially available, which is 99%. And so you still have protons in the other pool, in the CH pool. Now, what we can do computationally is we can recalculate the spin dynamics, but only include proton pairs and clusters with up to a certain maximal proton-proton distance. And this will help us to figure out which proton pairs actually contribute to decoherence. So here, this is the cutoff plotted horizontally. And vertically, I'm showing the inverse of the phase memory time. It just makes a more beautiful plot. So up to 1.5 angstroms, there is no proton pairs. But once you hit the water distance, you get a small jump, telling us that the proton pairs on water contribute a little bit to dephasing in both samples. Once we hit the HCH distance, the sample where the glycerol has all the protons on it sees a massive increase in the rate, decoherence rate. And then the alpha and the beta proton pairs don't contribute that much anymore. In the OH sample, where we have a lot of protons in a water pool and still some protons in a glycerol pool, we'll see that the water contributes little and the glycerol actually still is the major driver. And the reason that is the case is that, whenever you have a glycerol molecule with 1 proton, you have a glycerol molecule actually with 5 protons, right? So there's always a cluster of 5 protons present. So the correlation of protons is absolutely crucial in determining phase memory time. Quite an interesting experimental finding here too. I'm kind of running out of time, but let me just briefly mention in that 2 slides or 3 slides what happens if you take these decoherence examinations further to the refocused hand echo. And the reason we want to do this is because spectroscopy, the measurement of spin-spin distances, uses a refocused two-pulse echo. Here's an observer frequency, and there's a pump pulse. And as we sweep the pump pulse in Deer spectroscopy, we observe a modulation, and the modulation frequency is modulation period is proportional to the distance cubed. And so, of course, in order to measure long distances, we'll have slow modulations. We need very long tau2 values, making it very challenging to see the electron spin echo if the decoherence is bad. And so here we collaborated with Daniela Goldfarb because she saw some interesting effect and she thought we might be able to explain them. So if we just strip the pump pulse from the Deer sequence and just look at the refocused Han echo here, this is kind of the simplest form of a dynamic decoupling. And we measure the echo amplitude as a function both of tau1 and tau2 in a two-dimensional plot here. So tau1 horizontally, tau2 vertically. You can, of course, see that if you keep tau1 and tau2 short, you get the most intense echo here. And as you increase them, tau1 and tau2, it decreases. Now in Deer, we are required to pick a certain tau2 because we want to observe a certain dipolar oscillation frequency, right? So let's say 3 microseconds. Now, what is the best tau1 to measure? Well, it turns out it's not 0, so which would make the overall sequence shortest, but it's also not equal to tau2, which would be the Carr-Purcell dynamic decoupling condition. Turns out you have to pick a tau1 that's in between to get the maximal echo amplitude. And just to shortly summarize, we can recapture this with simulations, basically quantitatively. And if you dig into it, it turns out to be a 3-nucleus effect. So it's not just proton pairs that have observable and practical consequences, but also the effect of proton triplets. With this, I want to briefly conclude and say that it seems to be possible to quantitatively predict electron spin coherence with molecular dynamics and quantum dynamics. The effect, which looks like relaxation, is actually just unitary evolution in a really large spin system, statistically averaged over a lot of configurations. The main drivers of phase dephasing are protons that are 5 to 7 angstroms from the electron spin, and proton pairs that have distances that are less than 3 angstroms. And there also we observe effects that are beyond proton pairs, but also involve triplets. Of course, then the impact of this, of course, is super cool to understand is fundamental spin physics for these many spin systems, but it has practical relevance for proton spin labeling in Deer. And also the chemistry qubit folks are excited about this because it would help them, it can help them design longer coherent systems. I want to thank Lizzie and Sam who did all the work and all my group here at an ice skating outing a couple months ago. And I'll thank you for your attention.
Okay, well, thank you for a really very incisive talk, Stefan. And there's one question already from Ilya. Well, 2 questions. All right. Is Rinbao's version of cluster correlation expansion massively parallel? Do those 500 million clusters need to talk to each other during the time domain evolution? If not, GPU is your best friend. That's Ilya's question. All right.
Yes. No, these cluster, basically what we need to do is for every 2 cluster and every 3 cluster, we need to solve density matrix evolution completely and explicitly, right? But this is all trivially parallel. And we're using MATLAB with GPUs, GPU arrays to do that. And sometimes we manage to get it to run.
Yeah. And then Jeff Reimer says, good morning, Stefan. An analogy to solid state NMR, can one imagine EPR pulse sequences that select cluster sizes and then measure their decoherences? Okay.
Yeah. Good morning, Jeff. Yeah. So yes, we would basically have to do some proton NMR work on these clusters. The problem is that they spectrally overlap. So I don't think it's possible to individually address specific clusters because when we're in a broad matrix of protons, they basically all have the same resonance frequency, differing by kilohertz level, which is beyond what EPR equipment can resolve.
And Walt Masewski asked, have you measured the impact of the exchangeable protons on decoherence as a function of sample temperature?
No, that's actually an interesting question. So we have not looked at temperature dependence. Well, we have looked at it, but only to the degree to assure that it's not temperature dependent. So as we decrease temperature below 50 Kelvin, the phase memory time plateaus. And because all the other decoherence mechanisms are quenched, and as we start, if you would go back up in temperature, what would happen is like methyl groups would start to, dihedral bond angles would start to librate, or dihedral angles would librate, methyl groups would start librating, and everything would soften up and add new decoherence mechanisms that would take a lot more effort to model properly. So we're staying specifically in this low temperature limit to isolate this one nuclear spin-driven electron coherence, decoherence effect.
So Klaus Schmidt-Rohr asked, beautiful presentation. Did you measure the residual proton concentration in the 99% glycerol? Was it 99% glycerol, or was it 60-40 glycerol, or what was the glycerol concentration? I don't think you said
the glycerol, I didn't say that. It's one to one by weight.
So 50-50 roughly.
Yes.
You seem to say that a residual protonated glycerol is fully protonated. Did I understand that right? Okay.
Correct. Yeah. If you buy, basically, if you get you can get just glycerol, either D3 or D8 or D0 glycerol, right? So the CH protons are either all hydrons, are all protons, or all deuterons.
Mm-hmm.
So there is no kind of scrambling that's happening there for the CH hydrons.
So Fred Mintic, who's the DMP person, asked, have you tried D6 DMSO instead of glycerol? That D6 DMSO, deuterated DMSO water in a sort of 60-40 up to about 80% DMSO forms a nice glass as well, a stable glass.
No, that's an interesting suggestion. No, we have not looked at that. Our main motivation for this matrix was that it's the matrix that's relevant for protein DIR measurements. But D6 DMSO is an interesting thing to look at. I agree. Thanks, Fred.
Yeah. And it's the same. Okay, there was another question that popped off here. Is the same proton-proton flip-flop responsible for the T1E relaxation?
No. The proton flip-flop, if you just think about the time scales, the proton flip-flop is on the order of tens of kilohertz term, and the effect we see is on a microsecond time scale. The T1 for these electron spins at low temperature, it can be many milliseconds. And what drives T1 in these environments is typically either phonons from the bath, or it's mediated by spin-orbit coupling, which are two of the more common T1E mechanisms in this regime.
And Lucio Friedman wants to know, how do you ensure that relaxation, even nuclear relaxation close to the electron, plays no role? And two, he wants to know, what's the origin of the stretched exponential relaxation you mentioned?
Lucio, the second question is a very interesting one. The first one, I think we didn't measure nuclear relaxation directly on those few protons that would be nearby. Yeah, well, to say we don't have experimental evidence for that, but so basically but what we can do, for example, we can compare the D18 tempo with the H18 tempo. And besides these ECM effects and methyl effects, we don't see a stark difference between those two. It's kind of suggesting that even if those nuclei would relax on a time scale that's comparable or relevant, I don't think we would have a chance to observe this. So my conclusion from the data is that nuclear relaxation of these protons is not directly, so the T1 relaxation of these nuclei is not relevant here. So, but the second question is really, is indeed quite interesting. Where the heck does this, how can we connect the quantum spin dynamics to the stretched exponential. Right? And I have to admit that I don't quite have a clear picture of this yet, right? You know, there's older models in the literature that are able to derive this form from a stochastic kind of flip-flop dynamic flip-flop process where you have a proton bath and you take protons in pairs and you assume kind of like some sort of flip-flop stochastic flip-flop rate, and then you can do the stochastic averaging and end up with a stretched exponential. But how we do get from the quantum dynamics density matrix evolution to a stretched exponential form is something I have to admit I don't directly understand yet.
Yeah. Okay, so Thomas Schmidt, it's great talk, Stefan. Quick question about the proton. One could easily explain from that data the findings by the Eatons that methyls drive the decoherence. Would there be a difference between 2 and 3 proton pairs?
We are working into including the methyl groups into this calculation, into the CCE expansion. And as we've seen, we do have data, and you know, the Yashka lab has extensively worked on this, as have the Eatons, understanding the methyl group effects. Let me pull up the slide again. Right. So if you look at, again, compare these two, you can see that there is obviously the black curve is a little flatter here, and flatter here, and then gets steeper again, indicating a methyl contribution. And in principle, it's possible to include this, but it gets very tricky because a methyl group is not going to only just couple within itself, but it's also coupling to the environment. So we kind of have to couple tunneling dynamics to these flip-flop terms with other protons as well. So we're working on this, and hopefully we're able to incorporate these effects in the quantum dynamics simulations very soon.
Gunnar says, very nice work. Did you try to differentiate between clusters of protons on the same and on different water molecules?
Let me pull up the slide. So we have not, you know, there's so many ways of slicing and dicing these simulation data. We have not done that, Gunnar, the way you suggest. But we do see that basically from these data here, we do see that even that the intramolecular waters don't make a huge contribution in the samples where we're looking at. And it would be my expectation that, you know, the dipolar coupling doesn't care whether it's intra or intermolecular. It's just distance dependent. And I think the intermolecular water HH distance is kind of buried underneath here. And in our data, it's unfortunately hidden by the glycerol peaks, and we just can't get glycerol that's purer than 99% deuterons. So it's kind of an experimentally challenging corner case to access to get explicit data on intermolecular proton distances.
So your slide here is saying that the glycerol geminal protons are really the ones that do the relaxation.
They do the damage, yes.
They do the damage, and that says that there may be clustering between the radical and glycerol.
Not necessarily.
Not necessarily?
No, because this is not a purely distance dependent effect. So we go back here. So you can see that the contributions, the contributions are relevant all the way out to about 10 angstroms, right? So we don't necessarily need to have a cluster of the glycerols attached to the nitroxide. And actually, we probably could look at our molecular dynamics frames and see if that's happening, right?
Well, the reason I bring this up is that Kong and I published a paper on the 3-spin solid effect a while back or so ago. And we seemed to see, what we saw was that the polarization in the DNP experiment was tridal. It wasn't nitroxides, but so it's a different radical. But the polarization did transfer first to the glycerol, then to the water. So there.
What was this in DNP juice, or what is the deuteration composition of that matrix?
Yeah, it was DNP juice, right? But we actually deuterated, you know, the water and deuterated the glycerol and so forth. But anyway, let's go
Yeah.
on to the next question and talk. So the last question, great talk. Have you applied the simulations to the Carper cell decay, and if so, do they fit well?
We have not. Others have. Kveter in Croatia. She has looked at this, and in solid state physics community PPF people have looked at this for NV centers and similar defects in crystalline solids. And what they find is that as you increase the number of pi poles in the Carper cell train, you have to include larger cluster sizes. And there's also an odd even effect in the sense that for an odd number, you tend to need smaller cluster sizes than for an even number of pi pulses. But these quantum dynamics methods do describe the Carper cell dynamics pretty well based on work from others.
Okay, and one final question just popped up. If the tempo molecule is fully deuterated, oh, another one popped up. Will it minimize decoherence due to the solvent protons?
From these measurements and calculations, I think that the deuteration, again, I think I need to go back to these data here. Right, basically just looking at the contrast here. So decoherence, those few protons that you have on tempo, they are too close in to the electron spin to contribute to these sort of proton pair decoherence effect. The only thing the protons do that they're close in, they contribute some ECM, right? And you can see some wiggles here in the simulation. And but there's too few of them, and their pair contribution is way too small because their hyperfine couplings are way too different. So this delta A is too large.
Okay, and one final question, and we should go to the next talk by Fred, is in your MD simulations, do you use a specific water type like TIP3P?
Yes.
The water rich,
Correct.
basically, do you allow the OH length to change?
No, we just use the fixed TIP3P water, and there's there's certainly room for improvement, and we think that our MD structures, they're probably not correct, right? But they're probably good enough. The proton density is kind of within a few percent of the experimental proton density, but I'm sure structurally the MD does not perfectly reproduce the experimental situation, but it seems to be good enough. And Fred, if you know better methods, I'm happy to learn about those.
Okay, well, that was again a very stimulating talk, Stefan. Lots of interesting new data. And Kong, do you want to do the next introduction to get us going?
Okay, thanks, Bob, for letting me know just a few seconds before I should do this. Okay, great. Thank you, Stefan, for a very nice talk. So I will introduce the second speaker, Rachel Martin from UC Irvine. So she started her undergrad in Arizona State University, and then after that, she went on to do a PhD with Kurt Zim at Yale University. After finishing her PhD, she did a postdoc with Alex Pines at UC Berkeley, and she's currently a professor in UC Irvine. And the title of her talk today is NMR Instrumentation for Everyone Using Additive Fabrication and Automated Benchmarking, which is 3D printing, so which we just talked about just now. So the floor is all yours, and we are very looking forward to listen to your talk. Rachel, please.
Great. Thanks, Kong. I'm happy to be here. So I'm going to tell you a little bit today about some of the work we've been doing with 3D printing and automating benchmarks of NMR probe development. And this is something that I'm really passionate about. It's something my groups are working on for a few years now. And I think it's, it's really a good way to improve accessibility of NMR instrumentation design for, so that more people in the field can get into it. I think historically, probe building has been a really specialized thing. There's only a few labs that really do it. And I think with recent developments in 3D printing and just cheap hardware that everyone can buy and use, there's really an opportunity to change that. So before I get into the details about building this stuff, I want to talk a little bit about what systems we're working on and sort of why we're motivated to do this. So why do we need new NMR instrumentation. Something that my lab is really focused on is these kinds of systems where it's not really a solid and it's not really a liquid. So that includes membrane proteins in a membrane. It also includes things like the transparent hydrogel in the human eye lens. So this is an amazing tissue that has up to 50% protein in the human lens, more like 70% in a fish lens, where the proteins have to do all the work of focusing light since there's no air-water interface at the cornea. And in these kinds of systems, we can really benefit from techniques developed for solid state and solution NMR. These tissues are really intermediate between what we would consider a solid and a liquid. So if we in the lens, for instance, we have this hydrogel that is, it behaves like a really stiff agarose gel. You can kind of pick it up and wave it around. But in terms of the NMR signal, it behaves like a liquid, up to really high concentration. So if we just look at HSQCs, this is just taken in a normal solution probe, up to about 250 milligrams per milliliter of protein, we can see most of the peaks in just a standard HSQC. And then the line widths start to get broader as we increase the concentration even more, approaching what we see in the actual eye lens. And so in that case, we need to do magic angle spinning. But, we're also really worried about homogeneity and, because these samples are quite mobile. And so we can benefit from making some custom coil designs. So in my group, we use both sort of standard solenoid coils, and then we also have some double saddle coils and other transverse coils that we use for different kinds of experiments. So we're really doing combinations between solids and liquids experiments, sometimes on the same sample. Okay, so to start off with, I'm going to talk about some of our efforts to make solenoid coils more reproducibly. So this is kind of the standard MAS probe coil. When I was in grad school, Kurt Zelm taught me how to wind them on a Phillips head screwdriver. And I found this amazing that, that's how it worked. And, even at that point that you have this, this coil, it's the heart of your NMR probe. That's what delivers the pulse sequence and listens to the signal that comes back. And it was made in this really low-tech way. You can do pretty well doing that. You can make really nice coils, but it's hard to teach people how to do it. It takes a long time. There's a lot of practice. So you can wind coils by hand and get really good RF homogeneity out of that. This is a nutation curve for a hand wound coil. But, if you have novices doing this, you can do the same thing and get really poor results. And so what we wanted to do when we started out in this direction is to make just this process of making the coils more reproducible. And so the idea was to start with a CAD program, and even, start with finite element modeling of what we want the fields to look like, and design our coil, and then go from that CAD drawing to a 3D printed coil. Now, 3D printing of metals exists, but it's at least from what we've seen so far, it's not quite good enough to make something like a signal coil for an NMR probe. I know Peter Gorkov has been printing trap coils and things like that, and it is improving. So hopefully it'll get there soon. But there's still problems with resistance. You get little air pockets in there. It's not, at least, in our hands so far, it's not quite good enough to do what we need to do. And so, so we've been looking at other methods. Okay, so I want to mention, where do we get these coil designs? This is a collaboration between my group, that led by Jessica Keltz, and Robert Morosi, who is in Filippo Capolino's lab here at UC Irvine. And here we're looking at what's the ideal solenoid coil, at least in terms of the axial homogeneity. And so Robert set up a simulation in MATLAB where we can parameterize the kind of the features of a solenoid coil. And so what Robert is doing is searching the space where the parameters are things like, the pitch, the spacing between the wires, and then also, we don't have to have even spacing. We can make variable pitch solenoids. Things that are constrained are things that are dictated by our stators, the spinners that we have. And those parameters are the axial length, the internal diameter, which of course is dictated by the rotors that you want to use, the wire gauge, and then also the minimum pitch between the turns, which is, we don't want to have them touching each other or close enough together that we have arcing. And so these are things that are just dictated by what probe are you going to use and what does the spinner and the rotors look like. And then we also have some practical constraints. We don't want to have a huge gap in the center, and we don't want to have the thing wrap back on itself. And so if we set up this parameterization, which Robert and Jessica have done, we can search this whole space of coil pitch and look at things all the way from just the constant pitch solenoid to sort of more extreme variable pitch examples, and optimize what we want the field profile to look like. And so in this case, what we're optimizing for is just having the maximum length of a nice flat field profile in order to be able to fill up the rotors as much as possible. So for a lot of these experiments with the crystalline proteins, we're using 3.2 millimeter rotors instead of smaller ones. And that's a deliberate choice because we spend a lot of time optimizing protein expression. And so we can actually make more than 100 milligrams of that protein per liter of E. coli. So having the sample is not really a constraint. We want to be able to fill in as much as possible. But since these are liquid-like samples, we want to get as much homogeneity as possible. So we're optimizing for being able to fill up the rotor as much as we can and still get decent line widths out of these pretty unforgiving samples that are mobile. Okay, so by using these coils that we've optimized, using Robert's program, we're able to make these, ideal, at least for the parameters that we're testing, variable pitch solenoids. And it gives us a pretty significant increase in how much sample we can fill in, while keeping the field above the 90% threshold. And so, you can see some differences here. We're still above this 90% threshold, but we can, this one with a little bit larger gap in the middle, has a dip in the middle, whereas, we can get one that's almost the same volume that's much more flat. Which one's better depends what you want to do. And that's, we can talk about these trade-offs at the end if people would like. Okay, so how do we make these things? All right, so we got through the first step. We optimized it in MATLAB. We figured out what the field profile we want is. And then how do we actually go about doing this and getting it to happen reproducibly? So the thing that Jessica came up with, we're not up to 3D printing the metals at this point, but we can 3D print a template to actually produce these coils pretty quickly and reproducibly. So she 3D prints the CAD template in ABS, and it has little grooves that you can use to wind the wire and keep it at the right spacing. And notice it is variable pitch. And then we can wind these things on the templates, and we can actually put our little Teflon coil forms on there, while it's already wound on the template, and and keep it, keep the ends and the ends of the leads in the positions that we want. And then ABS is soluble in acetone. So we can just throw this thing in a tube of acetone and let it sit overnight. And then the next day you get your finished coil that looks like this. And this is really nice because it enables us to make the same coils over and over again reproducibly. The thing that we still need to work on is that if Jessica makes the coils, they're awesome. And if people who haven't done it before make them, it's still a little bit hard. And so I think what we're finding with some of this, with, with the 3D printing and, trying to make this technology easier for everyone to use, we're finding that we have to be really careful with our documentation and our instructions on how to actually do it. And I think that's something that we should pay more attention to as a community than we do. I think a lot of times when we write probe papers, the audience is assumed to be, Bob and Lucio and Songyi and other people who build a lot of stuff. And we should be trying harder to reach people that, that don't build a lot of stuff, but they want to try it. And so that's something that we're striving to do in our current work. We're trying to be more detailed about the procedures that we provide for making these things. Okay, so how do our templates work? So we do see, that we can make these things reproducibly, and we can get these these different field profiles across the coils. So when we published this paper a couple of years ago, we made a really exaggerated one with a big hole in the middle so that we could really see the field dip. We were worried that we wouldn't be able to tell much of a difference between these things. Is, if you look at them, they're not, they're not actually hugely different from each other. And we were able to measure differences in these field profiles, and the simulations do predict what they look like pretty well. And so we're doing this measurement using a ball shift assay, which is that's something that's been around for a long time. Lots of people use this in the field. In this assay, we have a little conductive ball, little piece of metal that we move through the coil, and then we measure the displacement and the frequency, and that gives us a nice way of mapping out the field profile on the benchtop without having to take spectra of it. And so if we do this for our various coils that we made using the 3D printed templates, we see that we can reproduce the simulated fields pretty well from our little 3D printed template. So we have the one, where there's more of a dip in the middle, and then we have the one that's more flat. And then of course the stretched version that has a huge gap in the middle, the field drops down like we would expect it to. It's not as symmetric as maybe you would like, but we can reproduce the field that we were expecting to get. Okay, so for our parameter that we were optimizing for, which is just how much of this length can we pack with sample, we did pretty well in terms of being able to increase that and fill up our rotor with our crystalline protein. Okay, so I mentioned that this is not necessarily the most trivial thing to do. Again, we found that if an experienced person makes these things over and over again, they're really reproducible. They come out better than doing something by hand kind of in the same amount of time. But if we get inexperienced people to do it, there's a lot of slop here. And so, again, what do we do about that? One of the things that we initially tried is trying to make the template more foolproof. So making the grooves deeper so that you're really sure where the wire goes, and it's a lot harder to wind it the wrong way. But then there are trade-offs associated with that too. So if we start making the template too fussy, we have huge cuts in they're very deep cuts in the template material, then you start to get failures that look like this. And so I think there's no way to get around having to have very clear instructions for how to make these things. All right, so that's how we're actually making the coils and kind of our new paradigm for how we want to build stuff. We're trying to get to the point where we can 3D print a whole probe. Obviously, we're not there yet, but we're trying to do as much of it as possible. Okay, so having talked about that, I want to get back to this ball shift assay, because that's another thing that is it's a fiddly little thing that you have to teach somebody to do, and it's kind of hard. So the way this works is we're moving a little conductive piece of metal through the coil, and that's going to perturb the magnetic field. And we measure that as a frequency shift. And so we can do that, using a network analyzer. It's pretty convenient because we can do it on the bench, and it gives us a magnetic field profile. And that way we don't have to waste magnet time on, measuring the fields of coils that aren't very good, especially, and this is especially important for things like testing reproducibility. If you have 10 of them and you want to see if they're all the same, it's pretty annoying to have to keep changing the coil in and out of the stator and actually taking spectra of these things. So it's useful to have benchtop tests. However, at that scale, when you have a bunch of these things, even that little benchtop test gets to be, pretty time consuming and difficult. So the way we usually have it set up, we have the little ball mounted on a fine threaded screw that we can move through the coil. And, you have to be careful how much you turn the screw, and, it takes some practice to be able to do that well. And Jose Uribe, who's a graduate student in my group, wanted to speed this up and automate it. So he built a little apparatus that can actually do this measurement by itself. And this thing, now that we know how to make it, costs less than $150 to put together, and takes some time to assemble, but maybe about 8 hours. And once you have it, then the time to measure each coil profile is a few minutes, and it has improved our resolution greatly, by about a factor of 10. And that's because we're going from just having a finely threaded, 440 screw that you turn manually to push through the coil. Now we have a stepper motor that's, that has very finely controlled position. And so now what we can get to is there's just a trade-off between how long do you want to wait for it. So you can get this thing to move, really slowly and take a million data points, but then it takes a long time. But it's all automated, so you can set it up and go have coffee, and it works. And this thing is also pretty easy to build. It opens it up so that even probe building novices can now test these things on the bench and see, okay, do you really need to ship your probe in for repairs, or is your coil okay? And for people that build stuff all the time, it's it makes it much easier to test a lot of these designs quickly. And so one feature of this that I want to point out is that we're using this with the little nano VNA, portable network analyzer, which is pretty amazing. I mean, so you can buy these things on Amazon. They're like $80, and it just, measures the frequency, and you can output it to your computer. Jose wrote a Python script to collect the data automatically, and it's really easy. At this point, you could just hook it up to a cheap laptop, and it will dump the data output in there. So the actual circuit that controls the ball shift apparatus has an Arduino microcontroller, and there's a stepper motor that's attached to the little actuator that pushes the ball through the coil. And this is the particular chip that we're using with the Arduino controller. And if we look at, how does this thing do with the positional accuracy? If you try to get it to come back to the same place over and over again, it's really great. And then if we measure the angular deviation, it looks like my data didn't show up on that, but that actually comes out really good as well. So at this point, this ball shift assay is completely automated. The the little device that does it is much better than a human at measuring these field profiles, and we can do it really quickly. And so that enables us to speed up this whole process quite a lot. And so now, we can prototype our stuff using CAD files and, do some of our troubleshooting before manufacturing. So we can look at, some of the problems that, that we might be able to foresee, in the CAD file already. And then, we can go to optimizing the 3D printing. And that's something that I want to spend a little bit of time talking about, because a lot of people don't. So a lot of times you hear people talking about 3D printing as though you just push the button and your object comes out. And if you're lucky, that sometimes happens, but not necessarily. So there are a lot of parameters that you have to set up with the 3D printer. And so we're mostly talking about fused filament fabrication, for the stuff that I'm going to talk about today. There's also stereolithography. We've worked with both. There are advantages and disadvantages. But it turns out that there are a lot of parameters that you can optimize and change and mess up on a 3D printer also. So it's considerably easier than learning how to do, really advanced machining, which I have done. I spent a lot of time on that in grad school. This is definitely a step forward, but it's also not completely trivial. So these are things that I'll talk about a little bit later. But so if we look at our what we get from the automated ball shift measurement, these are for some of our better variable pitch coils. I mentioned that there's a trade-off between how fast you make the measurement and how many data points you get. So how finely do you map out the field profile? So if we put our little stepper motor in full step mode, this is the density of points that we get, along the rotor axis. And then, we can go into half step mode. So, a factor of 2 more points, and we can really start to fill this in. And then, we can do it again. And so this really gets into, it takes a long time to measure it. Our comparison to the CST model is over here. And, what's the best thing to do? It just depends what you're doing. So, if you have a bunch of these coils and you just want to see, are they good enough? Maybe this is fine. If, we have a new type of coil design and we're interested in whether it matches the model, and especially if we have reason to think that there are going to be, some interesting differences in here, then we can map it out really finely. It's just, how much time do you want to spend? But it's great to have the flexibility, and also to have this automated system where the measurements, can be done very quickly. And so then just to compare, this is the same thing for the constant pitch coil. So, we can see the field profile isn't as flat. And one thing I want to point out, I know there are some people in the audience who are likely to point this out. So here, both of these plots they're normalized to the highest field strength. So for the constant pitch coil, depending on what you're doing, it may be possible to get more signal by restricting the sample, because this overall field value is higher. So that's something that we can talk about more in the discussion. Here we're mostly focused on, can we see the differences in these field profiles and, map this out with our automated system. Okay, so we can also do other little testing modules with this, with the nano VNA and these automated systems. So Jose also made a little circuit that reads the value of capacitors. And so here, he's testing this with a commercial capacitor that we knew the value of. But this is really valuable to us in our lab, because we make a lot of homebrew probe parts. So a lot of our probes use transmission line elements. We make capacitors and inductors out of little pieces of transmission line. And, of course, you can calculate what the capacitance and inductance is supposed to be, but it's convenient if you can actually measure this on the benchtop. And so this is something else that we can do with these little Arduino systems. Okay, so the last little bit I want to talk about is some of our adventures with 3D printing fluoropolymers. So far, what I've talked about are things that, that you can print in ABS, the inexpensive polymers that come with a 3D printer. And you can do a lot with that, but there are a lot of pieces of an NMR probe that, that are sensitive to what kind of material you use. So obviously, we don't want protons everywhere. Some of these polymers have very lossy dielectrics, and so you wouldn't want to use them for any of the components that are actually involved in the electronics. So even though they're really useful for structural things, we're not necessarily able to make the electronics that way. And so in order to do that, we really need to get into printing fluoropolymers. And so we started out with this, looking at stabilizing the coil assemblies of our cross coil probes. And so I'm not going to talk about this probe very much. I just want to point out that it's a cross coil assembly that has, a resonator on the outside and then a solenoid on the inside. And, of course, lots of other groups have built cross coil probes. We've been inspired by a lot of their work. And we've been concerned about how do we keep this assembly from moving around? How do we keep the coils stabilized relative to each other? And so one of the things that we want to do is to have a PTFE coil insert in between these two coils. That keeps them stabilized relative to each other. It helps prevent arcing. There are a lot of good reasons to do it. But the insert design that we wanted to use, like to maximize the filling factor of the coil, is too difficult to machine. So machining PTFE is pretty hard anyway. And then making these tiny little thin cylinders is just impossibly difficult. They fall apart. I mean, you can kind of do it a couple times, but it's a challenging thing. And so we initially started working with 3M, and Per Nelson was our collaborator there. And they 3D printed some PTFE parts for us, and they worked great. So it's not just these little coil inserts. We did a couple of other things too, but then unfortunately they discontinued that program. So, so what are we going to do? So we have started experimenting with doing this ourselves. And so the thing that we're testing it with is the MAS and SAS spinning assemblies. So making those little coil inserts is, maybe not the thing to start out with. It's challenging just in terms of, how unforgiving it is. And then also the electrical properties are extremely important there. So we decided to start out with spinning assemblies because that's something where we could really benefit by having, even reduced proton background. And, it's not as important that the electrical properties be perfect. And so we're trying to 3D print our MAS spinning assemblies, and we've played around with printing the bearings also. And so this has the advantage that we can do things like we can use computational fluid dynamics simulations to simulate the airflow in our stators and make sure that it's doing what we want. And again, this is important for the types of systems that we work on, because sometimes we want slow magic angle spinning. So we don't necessarily want to spin really fast. Sometimes we want to spin slow, but we want it to be really stable. And the commercial stators are not necessarily optimized to do that. And so now we can model the airflow, and we can figure out how that modeled airflow correlates to the actual spinning properties that we want. And so the last little bit, I just want to show some details about how that works, because again, it's not as push button as we would like. And so this is Jose's work again with PVDF. And so, every time that you go to print in a new material, you need to optimize some things. So, so things that we can play with are the temperatures of the nozzle that's actually extruding the stuff, and then the bed that it's that it's being laid down on, and then also the overall chamber temperature. And modifying, all of these temperatures will give you really different results in terms of how the polymer comes out. And so, what Jose is looking at when he's going through these, these parameters is the adhesion of the different layers to each other, and what's the what printing speed do you need to do. So again, there's a trade-off. If you print it really slowly, sometimes you get better results, but it also takes forever. And then we have to be really careful with the nozzle temperatures so that it doesn't clog. And so then, once you have these big things, more or less working, then you can correct for other parameters. So like the extrusion rate, and then, you can do this until you get a smooth surface. And so in order to test these, there's this little model called Benchy, which you can look up online. It's really useful. It's a little toy boat, and it has all kinds of curved surfaces and holes and things that stick out. And so you can print this thing and see where it fails, and that tells you about how your setup is working. And so it's it's really nice because it gives you a lot of different mechanical features to look at as you see how to optimize your print. So here's what some of these things look like. So you can see there are different levels of, you get a rough surface on the flat parts. You can get these kinds of defects in curves, and then you can get holes in the thing if it's really not working well. You can see there's a gap here. There's a couple more holes over here. But if you get all these parameters optimized, your little toy boat looks good, then then it's useful to start doing other things. And so we can start to actually get useful spinning assemblies out of this. So here are some 5mm spinning assemblies that Jose printed, even without post-processing. So one of the things I think is amazing about this is when we first started this, I said, don't even bother trying to 3D print tapped holes because it's not going to work. It actually kind of does. I mean, they're better if you run the tap through them afterwards, but you actually can get to the point where you can 3D print a tapped hole and stick a screw in it and it starts to work. So, so far we've tried this for 5mm and 3mm spinning assemblies, and we're playing around with the airflow to different places to try to optimize for slow and very stable spinning. Okay, so I'm going to wrap up. I think, our conclusions are anyone can do this. It's not completely trivial, but, so we need those of us who are building things and trying to disseminate these ideas, we need to be really careful about providing good documentation so that more people can do it. But this does make it possible to just directly implement parameterized designs without worrying about how you're going to machine it, which is really freeing in a way. And, I think we can really improve this in terms of how we do the parameterization and optimization. I think there's a lot of room for machine learning and more computational work there as well. Okay, so, this is also my general message to the magnetic resonance community. We should keep building stuff. This is one of the great strengths of our community. It's a big source of our creativity, and I think it's something that we should really cultivate and encourage. Okay, so I think I'm about out of time. I would like to thank my group, particularly Jessica Keltz and Jose Uribe. They've done all of the work that I presented today. And then also Team Crystalline, I showed some of their work briefly. Brenna Norton Baker, who graduated recently, Megan Rocha, and also Marquise Crosby. And funding NIH and NSF mostly for this, and then also the Champ Opportunity Award to Jessica and Robert. And thanks for your attention. Happy to take questions.
Okay. Thank you very much, Rachel. All right, for a really nice presentation. Lots of new twists and turns on making coils and so forth. So you did a 3.2 coil, all right?
Mm-hmm.
Have you tried a 1.3 or a 0.7? Okay.
Not yet.
I mean, so they are all, I mean, as far as I know, wound pretty much uniformly. I mean, if I look at them under a microscope, yeah. The commercial coils, okay.
Yeah, that makes sense. So we haven't tried that yet. So partly because it's hard, and partly because for what we've actually been trying to do, we're mostly interested in these things where we can make buckets of the protein, and we want to use the big rotors and fill it up as much as possible. So like for that crystalline protein I showed, it's very mobile, so we don't need to spin really fast. It's better to have a lot of sample and to spin very stably. So that's why we've been more focused on 3.2 and 5 millimeter rotors. But yeah, I'd be really interested to see how it works for the smaller ones as well. We just haven't done it because you can't do everything. Art Palmer wants to know, does the modeling of the coils include the effects of the leads? And okay, as far as I know, no, but Jessica, please correct me if that's wrong. I think Jessica is here. But you're right, Art, that's something that definitely has an impact.
Okay, Walt Masevsky asked, how important is the stretching force you put on the solenoid coil wire when you wrap it?
That's a great question. And I think that is, I didn't show spectra from these things, but I mean, I think that is one of the factors that that can influence how well these things perform. And so if you want it to work really well, you anneal it. So we have, little furnace, and you can put it in there on the PTFE coil holder and anneal it. And I think it's less important for the solenoid than for some of the other more complicated wrapping strategies that we use. But for that, it's absolutely critical to anneal it. And again, when we get to the smaller coils, like Bob is talking about, that's probably more important there as well. When you have the thinner wire, you can probably create a lot more metal fatigue by just stretching it during the wrapping. So yeah, it's a great question. Annealing it is recommended.
Yeah, so so you've only used ABS for this, for the coil form?
No, we've tried all sorts of stuff, and ABS is what works the best. So otherwise we get into some of these things where you get failures pretty often, or the little grooves run into each other. So I think there are definitely other things you could do. In our hands, ABS is what works the best.
Dissolution and acetone, right?
Mm-hmm.
Yeah.
And that was the first thing we tried because it's cheap. Like that's what came with the printer, and also it dissolves in acetone, but it also turns out to work the best, at least for us.
So Art wants to know if you have susceptibility matched wire or plain copper.
We have susceptibility matched wire that we use once we get this optimized. And so that's it's palladium over copper. And so when we we just use regular copper wire for testing these things, but once we figure out what we actually want, yeah, we use the susceptibility matched wire. It makes a really big difference at 800 megahertz.
And Nick Alaniva, I really enjoyed the talk and the message. Have you looked at the performance of these 3D printed components at different temperatures? If so, how hot and how cold?
Yeah, so that's a good question. So the answer is we haven't really looked at it. I mean, we've gone from kind of, room temperature down to maybe 0 or so, but we haven't really pushed it. I think that's actually going to be one of the huge advantages of 3D printing, because there are all kinds of different materials you can use. You could imagine, if you want to make probe components for different temperatures, having the versatility to just make it out of different materials whenever you want is going to be great, I think.
Okay, Kong, do you have some questions you would like to add?
Yeah, just a quick question. So you mentioned like you were using a fluorinated polymer, and also like you're collaborating with 3M. Are these 3D printers commercially available? Or would you mind to name what are the printers that you were using to do some of this stuff that you show?
Okay, so the 3M thing unfortunately is not. So they discontinued the program. So they did the pilot project with us. and I'm sure other people too, where they tested 3D printing some of these components. And their idea for that is that it was going to be a service where you send in your design and they do it for you. And then they decided not to do it, and also not to make the printer available, at least right now. Hopefully they will in the future. The other one, which we've started playing with now, is just a commercial printer. And again, I'm going to ask Jessica, which one did we actually use? She did answer the question about the leads in the chat. I see that that the part that passes through the spinning assembly is simulated, but the rest of it. Okay, so it's the fun mat that we did that with. Thanks, Jessica. We have a whole 3D printer farm, and it's hard for me to keep track of which one gets used for what.
The printer farm, huh? Okay.
We do. It's really cool. We have a lot of stuff going on.
All right. Okay, so we've hit the 12:30 mark again. And so I think we owe both Stefan and Rachel a really big thank you for 2 wonderful talks this morning. I think they both generated a lot of questions, a lot of interesting new information, and we'll be going forward with that. Okay, I mean, we're doing a lot of 3D printing of stators, or we were doing 3D printing of stators, and so forth. So it's it's something that's really useful when you're trying to build a new probe. You can get a new part. Kong actually bought us a 3D printer before he left to go to Paris, and he wanted to take it with him, actually.
No, I did not say that. But I bought a new one.
He bought a new one. Okay.
And it took me a long time to convince you.
Okay.
Okay.
All right. So again, thanks to Rachel and to Stefan for great talks. And we'll be back here in 2 weeks. Oh, no, in 2 weeks, we have ENC, right? Yeah. Okay. So we will not be back here in 2 weeks, but we will be back in 4 weeks. So the next 2 weeks from now is the American ENC in Orlando, Florida. And we hope we will actually see some of you or all of you there. Okay. So thanks again.
Thank you.
Bye.